Lab 4, Animal Behavior Part 2, Territorial and Sexual Behavior. Acheta domesticus, or common house crickets, were observed under different conditions in order to study their territorial and sexual behaviors. They are sexually dimorphic, which means there are physical differences between the males and females. The female has a long ovipositor used for laying eggs. Males have an apparatus on their wings that allow them to sing. A cricket song indicates how large the male is. Sexual dimorphism leads to competition and territorial behavior. In this lab, 2 males were put into separate terrariums to establish his territory. Cohabitating females and males were then separated into larger terrariums by sex, according to their dimorphic structure. Then, males and females were added to 2 resident males' terrariums. The male crickets were unmarked, yellow or blue. The female crickets were pink or green. In the first container, a male was first added, then a female, and then another male and female were added. The marked males tended to be more dominant. In the second container, a female was added first. Then a male was added, followed by another male and female. In these cases, the unmarked male was always dominant. The behavior of the crickets were recorded for each contact made according to the 5 aggression levels. Table 1 is a description for each level. Level 1 is the least aggressive, while Level 5 is the most. The objectives for this lab are to study live animals in their habitat and record their behavior towards each other in the habitat. One feature of their behavior is studied in order to limit what is being observed. The observations are made using an accepted ranking system for territorial behavior, making it easier to share and understand data. The method for this lab was followed per the procedure of Lab 4, written by Mike Buckle. The 2 resident males were observed initially. For the first 2 territorial experiments, it was hypothesized that the resident male cricket will defend his territory by fighting with the other males. When female crickets are added, the males will guard her, mate with her, or fight with the other males. A male cricket was added to the first terrarium. In the beginning, both were aggressive towards each other. As shown in Table 2, one cricket usually retreated after fighting. The unmarked cricket was dominant. This supports a hypothesis because the unmarked cricket defended his territory against the marked cricket by fighting. This video is an example of Level 3 aggressive behavior of the dominating unmarked cricket. Next, a female was added to the terrarium. She approached the males and preferred the marked cricket. The unmarked male was the only one who chirped. He defended his territory and demonstrated ownership. Table 3 showed that the males followed the female around most of the time, but when there was fighting, they had a range of aggression. The hypothesis was not supported because the marked cricket was the most dominant and aggressive. However, both crickets were equally dominant towards the female. In this video, the female cricket approached the marked male. Then, the marked male cricket followed the female cricket. Another male and female were added to container 1. Table 4 shows that the unmarked male chirped around the females, while the other males guarded the females. The yellow male cricket was the most dominant. In this case, the unmarked resident male was not successful in defending his territory. The green female followed the pink female, and she followed the unmarked male. Then, a female cricket was introduced into the second terrarium with a resident male. The male followed and guarded the female, according to Table 5. After 4 minutes, there was little interaction between the 2. However, the male did chirp frequently, and the female followed the male. This picture is an example of the male cricket following the guarded female. Another male was added to the terrarium. There was little aggression between the 2 males besides chirping. When they did fight, the unmarked male was dominant and guarded the female. This data is shown in Table 6. Fighting did not solve the territorial disputes, but rather the males acknowledged the other's presence but stayed distant or close to the females. This is an interesting video because the unmarked male is mounted on the back of the female, while the marked male follows the female simultaneously on the left. For the last observation for container 2, another male and female were added to the terrarium. Table 7 shows that the unmarked male was dominant and followed the females. The yellow male cricket preferred the green female and fought with the blue male. The blue male preferred the pink female, and the pink female also preferred the blue male. Males tended to fight with other males over the females, but spent most of their time and energy following the females. In this picture, all the crickets are shown. They are separated by sex, with the females on the left and the males on the right. The final experiment was performed to test which substrate the crickets preferred. The 4 substrates were rocks, soil, cage bottom, and sand. It is hypothesized that the crickets, regardless of sex, will prefer the soil substrate. Table 8 is the cumulative time that each cricket spent on each substrate over 5 minutes. Both male and females preferred the soil. However, the male cricket also spent significant time on the sand, while females liked the cage bottom instead. Figure 1 graphically represents the total time each sex spent on each of the 4 substrates. Immediately, the graph illustrates that the female cricket preferred the soil, while the male cricket also preferred the soil, but was more accepting to other substrates. This data supports the hypothesis. The null hypothesis states that crickets do not have a preference in substrate. To statistically test the validity of the null hypothesis for the substrate data, an analysis of variance was performed. The resulting value is the p-value, or the probability that the differences in data observed are significant. If the p-value is less than 0.05, the null hypothesis is rejected and the data is significant. However, if the p-value is greater than 0.05, the null hypothesis is accepted. For the individual data, Figure 2 shows that the p-value is 0.0698. It is greater than 0.05, so the null hypothesis is accepted. That means that the crickets do not prefer a certain substrate, and the data was collected by chance. This data set was probably too small to accurately determine a substrate preference. The class recorded their total time spent on each substrate by sex as well. From this data, the average total time for each substrate was graphed in Figure 2. Similarly to the individual data in Figure 1, soil was preferred by both male and female crickets. Females greatly preferred soil, while the males only slightly prefer the soil to the other 3 substrates. The class data matches the individual data for substrate preference. For the class data, an ANOVA was performed as well. Figure 4 shows that the p-value is 0.0417. This is less than 0.05, and the null hypothesis is rejected. This means that the crickets do prefer soil as a substrate. Overall, according to the class data set, both males and female crickets prefer soil substrate. Over the course of this lab, crickets were found to be territorial. Male crickets defended their territory and demonstrated ownership by fighting, chirping, and mating. However, the resident male was not always successful in defending his territory and was not always the dominant cricket. Fighting did not always resolve situations. Most of the time, the crickets retreated from the others or chirped to assert authority. When the female crickets were introduced, the males tended to fight. However, there were more female-to-male contact than male-to-male contact. In the final experiment, both males and females preferred soil substrate.